So, this is our 10th mentor talk. So, just a reminder of the purpose of these talks: it's unity, critical thinking, service, community, and inspiration. And also, if you guys have any questions or suggestions, here's some information. If you guys have any ideas or anyone you know that you'd like to be a future mentor, feel free to let me know. And yeah, if you guys have any questions or suggestions or want me to add you to the Skype group chat, just let me know. So finally, we're welcoming our 10th guest, Dr. Farane Vercha Harem, and she is a cognitive neuroscientist, and she identified the FOXP2 gene, and which is also called like the speech gene, and I'm really excited to hear what she has to say about neuroscience and career, because I think like a lot of us here, I know Dr. Vetter invited her class, and so a lot of us here like are informed about neuroscience. So I think it'll be super exciting. So yeah, without further ado, Dr. Rahel Harem.
Great. Thank you, Jada June, for asking me to be with you here. It's lovely to see you all. So I was hoping to give you a little bit of a background first in terms of how I came to do what I do, and then to put it into context, and then give you an example of some of the work that I do, and then there'll be lots of time to answer any questions that you have. Does that sound like the right kind of thing that I should be doing?
Yeah, that sounds great.
Okay, I'll have to start sharing my screen. So let me open up the PowerPoint that I have. And then, so I'll just go to what I have to do. Do you see this?
Yeah.
Great. Okay. So I thought I would give you a little bit of a background, and it's a bird's-eye view of an academic career in developmental cognitive neuroscience. And the word developmental is very important because I deal mostly with children of all ages, from birth all the way to age 18, because that's where our hospital is actually seeing children up until that age. But in reality, once I start working with someone, I tend to follow them, and I've got patients that are now in their 40s and 50s, and I'm still seeing them because they've had their difficulties from birth onwards. So just by way of background, I wanted to cover a little bit about how where I was born and where I was raised, and the influences that I had, culturally speaking, linguistically, environmentally. Then a little bit about my education, and then my career track, and then I will end with a few words of advice. But then I'll be open to advice from you as well. So, well, maybe from my name you can't guess, but I was born in Iran, a beautiful country with fantastic, and it has relics of the past where civilizations grew and fell, lots of wars, lots of conquests, but lots of cultural evolution. And it brings it into the current times where there is a lot of contrast in every way, political, economic, social. So you have all of this mixed together. And as you can imagine, growing up in a culture like that can have its long-lasting influences. So I was born in a Baha'i family, but I was raised in a very strict Muslim country of Iran. And I attended a Catholic convent school. So I had three religious environments characterizing my development. And I would say that that's a very rich blend, because I was able to see from different perspectives the way of thinking that shapes the people that I was living amongst. Foremost, obviously, my family, Baha'is from many generations ago, but experiencing difficulties because of the Muslim community and the prejudices that exist against anything that's novel and new. And obviously, the Catholic convent school had its own advantages in many ways, because it was an Italian convent school, and it exposed me to the Catholic tradition, which was very attractive to me at the time when I was growing up. But later on, of course, I moved to the West with my family, initially with my family, but then later on my own when I came to Canada. But before coming to Canada, I was in a boarding school in England where I learned English. And then I returned to Iran for a few years, but this meant that actually my high school studies were interrupted and I didn't actually finish high school. But I had learned English and I was able to do quite a bit of work with the new language that I had learned. So I got a job when I was 17 and I was doing translations in translation of contracts with a Dutch consulting engineers firm. And it was very good to be able to earn a good living at the age of 17. I was very kind of unusual in that respect, particularly for a young girl in an Islamic culture to be working at that young age. And then after that, I decided that I wanted to go to Canada to be able to study at university. So this is where the long battle started, because of course I had not finished my high school and I didn't really have a high school leaving certificate to be able to enter university. And I was 17 years old and it was kind of tough to do this, to do what I wanted to do. However, the North American system being a lot more lenient than the British one, they told me that if I was able to sit the matriculation exams and pass them, then I would be able to enter university regardless of the fact that I had missed the good years between the ages of 12, 13, and 17. So the most, I guess you would consider the most important years of high school had been missed, but nevertheless, they allowed me to write the exams and I did that and I entered university. So I was living at the time in Canada, in Montreal, and probably most of you don't know, but Montreal has got a very big institute called Neurological Institute. And this is where some of the pioneers in brain research and brain surgery, namely Wilder Penfield, was practicing at the time. And it was always fascinating to me to be able to attend these lectures where these neurosurgeons would be talking about the treatments for epilepsy that they were performing by opening up the brain and stimulating different parts of the brain and ensuring that the regions of the brain that were the focus of epilepsy were going to be removed, but they wanted to make sure that the areas that they were removing would not be interfering with the cognitive aspects, particularly with speech and with memory. So it was amazing to me to sort of attend some of these surgeries where the patient would be brought in and they would obviously be anesthetized, and then they would open up the skull in order to remove the portions that of the brain that they wanted to remove. But before doing so, they would actually bring the patient out of anesthesia and then there would be this surreal experience of a person lying on the bed with their brain open and pulsating. And as they stimulated different regions of the brain, the person would actually be talking. So it was really, as I say, a surreal experience. You would never feel that you would be able to see something like this. So there was both a kind of amazement at what I was seeing, but at the same time, a desire to try and understand how this brain works, because it just seemed so amazing that you could see something like that. Anyway, that was the beginning of me becoming interested in brain function. And one of the people who really influenced me a lot was a professor who was alive at the time, and he was at McGill University in the psychology department, and his name was Donald Olding Hebb. Now, Hebb had also worked with Wilder Penfield, and he had been working with patients who had had frontal lobe injury, and he was trying to understand what the relationship between those regions of the brain that had been damaged were to behavior. So he had a lot of experience with patients, but he had also experience with monkeys, with animals, so that he could actually look at the wiring of the brain by studying animal behavior, but also human behavior because of the damage that they had sustained, mainly because of these wounds that they had received, or because of diseases, different types of diseases. So Hebb published a book called The Organization of Behavior, which actually has become kind of the Bible of modern neuroscience, because it describes how cells that fire together, wire together to form cell assemblies that then guide many of the fundamentals of learning of behavior and cognition. And I was fortunate that, along with about 20 other students at the time, we were able to sit in his lectures. It was called Hebb Seminar, and we were all in our early 20s at that time, and feeling very proud that we felt very capable in the sense that we had young brains that were working very well. And our pride was quickly washed away with Hebb saying that, well, as far as some aspects of brain function, you're already over the hill, because many of your immediate reactions and speed of information processing and working memory, all of those things are going down. But he reassured us that in due course, other things would be developing. Namely, he said, wisdom will gradually emerge, so that by the time you reach the age of 40 or so, you will feel as though you actually have accrued a certain degree of knowledge and wisdom. So we were very much kind of feeling disappointed, because at the age of 20, 21, 22, you don't expect somebody to tell you that you're over the hill already. However, it turned out that the advice that he was giving was very good. So he taught us two things that I remember very clearly, besides, of course, telling us how he came to his theories and how he was able to put the different strands of information together. Besides that, he gave us two pieces of advice. The first thing is that he said, you may be a scientist in one particular area, but you have to be able to talk to any scientist in any discipline. And the best way to do that is to be able to give talks in such a way that no matter which area of science your audience is from, they will be able to understand what you're saying and they will be able to follow it. The second thing that he said, so for that he said, you have to do interdisciplinary research. You can't just be sitting in your little office or lab and doing your own little thing without worrying about what the implications of your research is going to be in the wider world or in the wider scientific community. The second thing that he said is that you have to learn to give your talks in the time that you have been allotted. And he said that I'm going to teach you how to do this. So I will ask you, I'm not going to ask you to write any exams or do anything like that, but I'm going to ask you to give me 3 talks. The first one you have to do in 5 minutes. The second one you're going to have to do in 10 minutes. And the third one, you can choose the topic and you have to give it in 15 minutes. And I will sit and time you with a stopwatch. And if you go beyond the time limit that I've given you, you have basically failed. And if you manage to get your talk and the substance of your presentation across in the time limit that I've given you, then I will rate the quality of that presentation. So you can imagine that it was a very nerve-wracking experience to have a stopwatch in front of you ticking away and not really knowing whether you're going to be able to stick within the time limit or not. But that was a very good lesson to have learned from my professor at the time. So he had a profound influence because he obviously gave shape to the interest that had been awakened when I attended those surgical procedures that Wilder Penfield was performing at the time. So many years went by. I did a lot of my coursework and exams actually during the period that I lived in Montreal. And then with by that time, I had a family, I had married, and I had 2 of our 3 children. And then because of my husband's job, we actually moved to London because of his work. And I started looking around to see whether there was a place that I could actually work in the discipline that I had chosen and I had worked in. And I contacted another mentor who played a very important role in my life, and his name was Neil O'Connor. Neil O'Connor was the director of the Medical Research Council, and there was a developmental psychology unit at University College London at the time, and he was the director of it. And sometimes I really wonder how life works, because I had no idea who he was, and I had no idea that there was such a unit existing. And I had been given the names of a few people to contact, including a senior investigator who was in his department. So when I came to London and I was trying to see if anybody had a job for me, I called this developmental psychology unit. And at the time that I called, he was the only one who was there. So he answered the phone. So I said that I was calling and I had come from Montreal and I was living in London now. And I wanted to speak to his colleague because I knew the colleague worked with him. And he said, well, there's nobody over here, but what is it that you wanted to talk about? So I said that I wanted to work with children. I wanted to work with children who had brain injury. And I also wanted to work in a hospital setting. He said, well, I'm interested in that, but I actually don't work in that kind of a setting, but why don't you come and talk to me? So I said, okay, I'll come and talk to you. So I made an appointment and I came and saw him, and he said, I'm really interested in this. I can't help you. I don't have a job for you, but what I can do is to help you apply for a grant application with the Medical Research Council. And if you're successful, and I will support you to do that, then you can actually do what you want because you'll have your own money. So that's what happened. That's how I started. I applied for the grant with his support, and this is why the role of a mentor is so important. So he supported me in my first grant. And in those days, getting a grant, I was a postdoctoral fellow, basically. And with really nobody knew me in England at all. But the application that I put together was successful. And at that time, I got a sizable grant, which paid for my salary, a research assistant, and a half-time secretary. So I had the money, but I didn't have a position. And I wanted to actually go and work in a hospital. So again, it was Neil who supported me and found the right person in the hospital. And I ended up going to Great Ormond Street Hospital for Children, which is where I am still working at. So, and from all over the country, people, and actually all over Europe, people come to Great Ormond Street because it's a tertiary quaternary specialist hospital. So you get to see the children with different kinds of disorders that you read about usually in a textbook. You hardly ever get a chance to see such children. Anyway, I ended up at Great Ormond Street Hospital, and the sister institution is the Institute of Child Health. And there I started doing research, predominantly research in the first 5 years. And then gradually I thought that it was difficult to do research without having clinical contact with patients. So then I divided my job into 2 sections. One section was really doing clinical work, where children would be referred for diagnostic purposes to me and my team. And the other aspect of it would be doing research with those children that were of interest. So about, I would say that it must have been maybe 5 or 6 years after I had started working at the hospital that I got referred a family to identify, to be able to work with and identify the problem that they had, because this family seemed to have many children in it that were unable to speak properly. So I don't know what the background of most of you is. So maybe Jaydajan, can you give me a little bit of an indication of the audience that I'm speaking with? What is the background?
Yeah,
Are you in high school, or are you, some of you are in university, or what is the background?
so the ages from high school to university, I think most of us are in high school, and most of us go to Stanford University's online high school, the online school I go to. And so, yeah, most of us are in high school, and some people don't go to that school.
Okay. So if you want to interrupt me, by all means do. Do you want to interrupt me to ask me questions at this stage, or?
I think maybe we should just wait till the end, because then there'll be a lot more questions that might have to be related to this.
Okay. So Jada introduced the work that I do as some of this work that led to the discovery of the speech gene. So I thought maybe I work in several different areas. I still work in epilepsy, but I also work in another area of research, which concerns memory dysfunction and the amnesic syndrome in children. So that's unusual as well, but that has to do with memory. But I thought this one would be more interesting for you, so I decided to talk a little bit more about this. So this is really the study of the genetics of speech and language and how it relates to brain function, and the bigger picture of how it sort of opens up a window on how the ability in human beings to be able to talk has probably got an evolutionary basis, and it has evolved to become what it has become now. So I will talk a little bit about all of this. Anyway, so what happened was that I think it must have been around 1989, 1990, so a good few, about 30 years ago actually, that there was a conference held in north of England, and it was supposed to be drawing attention of the public to the plight of individuals who had difficulties communicating, using language for communication. And this is all forms of disorders that have to do with language problems. And many people from all over the country had gathered together at this conference, and I was supposed to be talking actually, but I was talking about epilepsy. But among the people who were gathered there, there was one of our colleagues who was a geneticist, And he was speaking at this meeting, and he was talking about the relationship between um, various genetic conditions and how these conditions affected the ability to talk in children. So after he finished his talk, there was a lady who was the principal of a school, a special school, and she came up to my colleague and she said, you know, this relationship between genetic disorders and speech and language is really very interesting, because I have a group of children in my school who have got the problem of difficulties with their speech and language. But the interesting thing is that they're all related. So obviously there's some kind of a genetic basis that's running in this family that's affecting the way they speak. So maybe you would like to study them and find out what is the particular gene that's responsible for this. So this family got referred to our institute, and then several colleagues became interested in trying to understand how a genetic disorder and the form of transmission of this gene actually was a Mendelian type of transmission, which indicates that it's a dominant gene that's doing this. It's being passed on from generation to generation. And with every pregnancy, if there's a carrier, if there was one person, a parent affected, and they run the risk that for every pregnancy, there's a 50% chance that that mutated copy of the gene would be passed on. So it's a very clear pattern of transmission across generations. And the family, which is really a godsend to geneticists, is the family is very large. So there were at the time 30 members in this family, and 15 of them had the genetic disorder and the other 15 didn't. So it was very clear that across 3 generations, the mutated copy had been passed down by an affected parent. So the family got referred to their institute, and my geneticist colleagues then asked me whether I would be happy to see the family members and be able to sort of identify what the phenotype of this disorder was in terms of their ability to communicate, and whether the phenotype could be used as a marker of dividing the blood samples that had been obtained from the family members so that the genetic analysis could be done in order to isolate the gene. So over the years, we ended up seeing about maybe half of the family members who were affected and a number who were not affected. And those who were not affected didn't have any problems at all. They were just healthy individuals. And the family members who did have the genetic disorder were otherwise very healthy. So it was only their ability or their disability in terms of their articulation. So we identified the family members. At the time, there was a grandmother, and then there were 4 adult members who seemed to have the same problem. And then each of the adult members had had a number of children. So the number of cases that we were working with was quite large. And that was very helpful to the geneticists, because there would be many exemplars of the same problem that they could test their the genetic component of. Anyway, so we identified the phenotype, and the phenotype was very well identified, in the sense that the blood samples that were obtained, it was possible to divide it. Maybe I can explain it a little bit better by showing you this slide. Can you see all of it? Yeah. So on the top, you can see the pedigree of the family. And the family pedigree, as you can see, you see these black dots and black circles and black squares. So everybody who's got a black color circle or square means that they are affected. They have that gene mutation. And everybody who's got a clear square or circle does not have the problem. So the problem appears to have started with the grandmother, who is generation 1. You see it at the center up here, right? And then she and her husband had 5 children, and only one of them did not inherit that mutated copy. But her other 4 children did. And each of them then had a certain number of offsprings who carried that same mutation. So altogether, at the time, there were 30 members, and 15 of them had these black circles and squares, and the other 15 were absolutely fine. So our task was to identify how, because they didn't have any other problems that we could see. You know, they could move well, they could do everything well. And in fact, they understood language not too badly, but they had difficulty expressing themselves. And what really was happening was that all the muscles that move in order to be able to produce these sounds using the lips, the palate, the soft palate, and the tongue, they weren't working properly as a result of this mutation of the gene. But we needed to be able to identify exactly who had the problem and who didn't have the problem. And the only way that we could do it, because there was a variation, some of them actually were not too bad. So we had to push them with different tasks in order to see at what point they would break down and whether that would then identify the core deficit, what we call the core deficit. So, but we kind of became aware of the fact that if these muscles are not working properly, then it's not only, of course, speech is the thing that becomes affected, but there must be other things that are affected as well. It's just that speech is so intricate and so exquisitely timed that you have no choice but to notice when something is not right. But for example, if your mouth doesn't move properly, nobody has got a measuring stick so that they can say, oh, that's an abnormal moving mouth. But the product of that mouth is a speech that is disordered. So then we looked at many tasks that were speech and language related, but we also looked at tasks shown here that do not involve any speech and language. It's just movement of this mouth, but in a sequence. So what is important is if what is produced by this region is sequential. So you can see that we asked them to, for example, copy, show me your teeth, stick out your tongue, purse your lips, and we found that they had difficulty doing that. So it wasn't only speech that was affected, rather it was an underlying ability to be able to move this apparatus quickly in time in order to be able to produce articulate speech. So our next question was, well, what does it have to do with the brain? Which areas of the brain are involved? So when we looked at the MRI scans of these individuals who were affected, we saw that there was really no outwardly sign of an abnormal brain. Their brains looked normal. But there was a technique that had just evolved at that time, which allowed you to be able to take the MRI scan and process them, and then use statistical techniques to compare voxel by voxel different regions of the brain and see whether each of these groups of voxels were showing less gray matter compared to the normal controls. So we used that technique, and then we found out, if you look at the left-hand side of the screen, you see that we found these yellow regions. And these yellow regions are regions that are part of a circuit. And that circuit is involved in movement of this region. It's also involved in production of speech. So if you look at the right-hand side, you see these two half views of the brain. And when in the functional MRI, when the patient or the healthy control is lying in the scanner and they are thinking of words and they're doing some manipulation with those words. For example, they're hearing a word like a noun, like telephone, and then they have to think of a verb coming from that noun. For example, telephone, ring, bread, cut. So you hear nouns, you have to think of verbs generated from that noun. When you do that, then in the normal healthy control group, you find that this region called Broca's area, which is well known for speech and language production, lights up in the normal healthy individual. But in the affected members, these two areas, which would be associated with that kind of activation when this task is being performed, do not show any activation. So it looks like they have less gray matter in these regions. And these regions are specifically developed in the human being to be able to produce speech. And if you have got less gray matter, it's an indication that the development of the brain, when it reaches that level of sort of development, has not fallen into place properly. And the reason why it hasn't is because there's been a mutation in this gene. So this gene has been responsible for setting up the circuitry that affects that top part of the brain that is involved in articulation. So this became really quite fascinating to be able to identify a gene that actually has got this kind of an effect. So this is the picture of the gene that you can see on the left-hand side. And when the blood samples were analyzed and when the genetic analysis was carried out, they found out that every single member of the family had a mutation where it's shown in that red strip that you can see there. Every single individual who was affected had a mutation at that point. Now, what is that mutation? That mutation, I think probably you've done biology, right? Okay, so that mutation was really a spelling error in the reading of the gene that substituted one amino acid for another amino acid. And that little substitution, which is effectively analogous to a spelling error, instead of a C, for example, it would be a K. But that was replicated in every cell of the body, and obviously brain cells, and setting up the brain circuitry. And that resulted in the mutation that had its effect in terms of their inability to be able to express themselves clearly. So the interesting thing, so the name of the gene when it was discovered in 2001 was really quite a discovery, because it was shown to be a control gene that controls the protein synthesis of many other genes further downstream. So it becomes a very important gene in that respect. And when it was found out, it stimulated a lot of interest to find out whether this gene actually existed in other species, and of course it does. It's the gene is found in all vertebrates. So from the lowly mouse to the bird to the chimpanzee to the human, it's the same gene, except that in our brain, it does what it does in terms of its expression in those critical areas that are subserving our ability to be able to communicate. So it's all a very interesting story. And it also raises the question, because we are so close to the chimp in terms of our evolutionary history, it raises the question of, if they have the same gene as we do, then why is it that they cannot talk and we can? What is it that distinguishes us? So this is when the next set of studies were done, and this was another collaboration that came about, looking at how this gene called FOXP2 has evolved over the millions of years, because obviously it sets up the circuitry for movement of all types. But as I say, it has undergone changes over the millions of years that it has evolved. So the changes that have happened between species in this gene are very few, but the more critical thing is that between the chimp line and the human line, how many of you know when our lines, evolutionary lines, separated? Do you know that? Can you guess when the chimp line separated from the human line so that we became humans and they remained chimps? Do you know that? It's estimated that we separated about 4 million years ago. So 2 changes appeared in this gene when the human lineage split from the chimp lineage. And those 2 changes were in this gene. I mean, there must have been other ones as well, but in relation to these, to this particular gene, 2 changes occurred in the amino acid configuration of FOXP2, and it raises the question of whether those 2 changes that occurred really are underlying the distinction that we human beings have in terms of being able to communicate with speech and language the way we do. So it's been a very exciting line of research, and obviously we started with behavior. We went to the brain, so we started here with behavior. We went to the brain, then we went to gene identification, and then to evolution, and we came back to behavior. It all happened because it was possible to do interdisciplinary research, which is what my first mentor taught us, to be able to talk to different scientists. But it also tells you something else, which is really important, which is cooperation and collaboration. Because without that, everybody loses. We would have never been able to do what we did in the end without the collaboration of the imaging scientists, without the geneticists, without the evolutionary anthropologists, etc. So everybody becomes a winner when you actually collaborate and cooperate with each other. And otherwise, you become very narrow in your science, and you become, and the benefit of it is not all, it's very limited for yourself, and it doesn't benefit others either. And the discovery of this gene has also opened up a lot of philosophical questions with regards to what it is that human beings have that enables them to be able to express their thoughts through speech and language, which is, of course, the fundamental characteristic of humans that sets us apart from all other species. So it has raised a lot of questions, not only about just the ability itself, but what it means to be able to express our thoughts, what it means to be able to express feelings about our inner core, about our spirituality, about our belief systems. All of that is tied in with our speech and language. It's tied in with our history. It's tied in with our evolutionary history, but also our identity and how we see ourselves and how we communicate with others. So it's really opens up many, many discussions, from philosophy to religion to economics, obviously to science, but it has a lot of implications, and it opens up the way to bring greater understanding among not only the scientists, but society in general. So I'm going to stop here and just say that the study of development is really critical, because not only we can see the formation of the individual from the very beginning to when they reach maturity, but it also gives us a lifelong perspective. Because if we concentrate our efforts on helping to the development of the young generation, it really fosters the development of a healthy society. Because if the young suffer, society suffers in the end. Okay, I'm going to stop here.
Awesome. Thank you so much. If people have questions, you can click on participants and then raise your hand, and then we can call on you.
Okay. Jada-Jan, you can't see my screen, can you?
We still can. I can unshare it for you.
Okay, stop share.
Yeah.
Okay.
Go ahead, Lexi.
Hi, so that was a really great presentation, and really, really interesting, especially since I personally am really passionate about like neuroscience and psychology and all of that field, and I'm planning on going into that. I was just wondering if there was one thing that you would change in your like journey to get here, what would it be? Would it be studying that last year of high school, or would it be maybe choosing a different path rather than doing stuff in epilepsy, doing something else? And also, how did you choose between neuroscience and psychology or psychiatry in that sort of field? Like, did you want to do research and kind of find out new things, or were you ever like leaning towards wanting to go down a more psychiatry route and trying to help people with mental illness?
Well, I must say that when I started, it was actually abnormal psychology or clinical psychology or psychiatry that really interested me. And in fact, neuropsychiatry, I think, is one of the most fascinating areas. The problem is that treatments are far and few in between. It's really difficult to treat psychiatric disorders. And in the end, I think it becomes a process of discovery with very little to show for at the end. It's getting better because many of the neurological disorders also have got a psychiatric component to them. So in that respect, I think it's becoming better and better in terms of treatments. But we're a long way away from being able to diagnose a problem and do something about it that is really life-changing and restores brain health and psychiatric health. That doesn't come about very often. And I think that, of course, there are many factors that contribute to psychiatric disorders. It's not just the genetic component or the predisposition of the individual or environmental factors. It's rather all of these things combined together. That's why it's so difficult to treat it. And I don't think that I ever really wanted to go down the route of psychiatry, even though I was interested in, as I say, abnormal mental processes. I didn't want to go down that route. I think I would have really liked to have gone down the route of neurosurgery. That would have been a very important route for me to follow. But, in the end, with the moves that we had with the family and having children, it just made it very difficult at the time. And now I don't regret it now, because I think if I had gone down that route, I probably would not have been able to get involved in these areas of research either. So I have no regrets. In fact, I consider myself very lucky. But I do regret one thing, and that is not having finished high school in the way normal individuals do. Because every building block helps. It was an uphill struggle for me. And I still suffer from some of the things that I should have learned when I was much younger, and I never did. So when it came to my my own children, I was so dedicated to making sure that they didn't miss anything, and they went through the process systematically and learned everything that they had to learn as appropriate to the age that they had at the time. And having been away from education for 3 or 4 important years, I developed such a hunger for gaining knowledge that I was in such a hurry to get back and try and make up for what was lost. But you know, it takes time to consolidate the knowledge. I crammed over an 8-month period something that was equivalent to 4 years. And yes, I passed the exam, but somehow it didn't feel as though all of that knowledge fell into place, because it never had the building blocks that it required. And that I do regret. So thank you for that question, because it does make me realize that I am missing something. But on the other hand, I gained a lot as well, so I can't complain.
Thank you so much. Yeah, that really helped.
Awesome.
So there are other questions, but Jada-Jan, are you going to be able to monitor the questions?
Yeah, I got you.
Okay, thank
I
you.
don't think anyone else has their hand raised for now, but earlier I think Matthew asked which amino acid was replaced. Oh, I think Dr. Vetter answered that. Okay, anyone have any other questions? Okay, there's a question in chat. I can read it if you like. So if FOXP2 gene that helps us with human speech development, how does the expression pattern of FOXP2 in humans differ from the pattern of expression in other animals?
That's very interesting, because I was just talking to a colleague about this earlier on today. In fact, when you look at gene expression in the mouse embryo and in the human embryo, the pattern of expression of the gene within the first few months of life, I mean, with human embryos, we can only go up to 3 months of gestation. We can't do research, gene expression research beyond that. But if you compare the first few months of gestation and its equivalent stages in the rodent, in the mouse, the areas of expression of FOXP2 are identical. But something happens, obviously, and this really tells you that it's not that species have not evolved. Of course, species have evolved. However, they are distinct. You know, you cannot say that a mouse turns into a human being. It doesn't work like that. A mouse is always a mouse, but it evolves within its own species. And even though the pattern of expression of FOXP2 and the areas where it's expressed during brain development is the same, but that expression does not mean that those regions do the same thing. They do something completely different, even though the neural tissue is the same. But the capacity is, well, there's no comparison, obviously. So that also teaches us something really interesting. A human being is meant to be a human being from the very beginning. And even though we share all the sensory modalities that we have, the 5 senses, we share it with all the animals. But out of those senses emerges something that is human specific. And it cannot be duplicated, even though the neural substrate of it is the same.
Thank you. So we'll have Matthew, and I don't know if Dr. Vetter had a question. I think her hand was up.
First of all, thank you very much for taking some time out of what I assume is your very busy schedule for coming to talk to us. I was wondering, what exactly do treatments look like, or are there any treatments for affected genes or affected areas of the brain, or is this kind of purely a sort of research and discovery at the current time?
Well, you're right that once the fetus is formed and the mutated copy it comes into being from the first cell division. So once that happens, obviously it's a done deal. However, having said that, we have been working with this family into their 4th generation, and we have been studying some of the offsprings from the time that they were babies to look at the evolution of this gene and how it affects them from the very earliest stages of development. Not that we can treat it, we can't put it back to what it should have been had this genetic mutation not occurred. But the very fact that we knew what we were looking for and how it was going to unfold as the development took place was a good guide to the parents in terms of how the early stages needed to be kind of monitored and shaped in order to enhance as much as possible the stimulation that would be needed in order to bring about as much improvement as possible. There are certain things that you can't work to improve because this is the way disability works. However, if you're aware of what's going to be happening, you can put things in place, both from an educational point of view, from an environmental point of view, in order to minimize the adverse effects of whatever disability you're dealing with. So you're absolutely right. We don't have treatments for these genetic conditions. Not yet. Certainly we don't have it for this particular condition. There are other ones where gene therapy can work, but this is not one of those things that we can work with yet. However, as I say, having as much information as possible is helpful because you know the trajectory. And if you don't do anything about the trajectory, you know that the endpoint is not going to be very good. Well, whereas if you are aware, you can put in things, particularly education-wise and therapy-wise, you can help to bring about improvement, but no cure. But for other things, you can. For example, on the epilepsy side, epilepsy surgery is very effective. It does actually stop the seizures. It brings about better quality of life. Sometimes it helps cognition as well. But nothing is without cost. Brain injury has always got a cost. It's just that you try to minimize the cost as much as possible.
All right, thank you. So I'm not sure if we have any other questions, and yeah, Matthew has one follow-up, and then I think we should probably conclude due to time.
Okay.
If you don't mind, I just one quick follow-up. Are there different severities? Like, does the gene kind of express itself with different severity in different people, or is the kind of effect without treatment pretty consistent?
You mean in those who are affected?
Yeah, and those who are affected, so those with the gene.
Yes, it does have different degrees of severity. In some, it really affects them very badly. In others, it's less affected. But the core problem is there. So as I say, the phenotype is there, but its expression varies. And over the years, we have seen there's this large family, but there are other individuals or there's smaller families that have been studied as well. And depending on